Hi everyone, this is Sahitya. I am here to present topic enzymes. Enzymes are biocatalysts synthesized by living cells. These are protein in nature, colloidal and thermolabile in character and are specific in their action. All enzymes are proteins, but not all proteins are enzymes. Catalyst is defined as a substance which increases the velocity or rate of a chemical reaction without itself undergoing any changes in the overall process. The functional unit of enzyme is known as holoenzyme, which is a combination of protein part and non-protein part or organic part. The protein part is called as apoenzyme and the non-protein part is called as coenzyme. The holoenzyme is also called as active enzyme. If the coenzyme binds to apoenzyme with weak bonds, then coenzyme can be removed easily through dialysis. If coenzyme binds to apoenzyme through covalent bond, which is a strong bond, then it cannot be removed easily with apoenzyme. This term is called as prosthetic group. There are two types of enzymes, monomeric enzyme and oligomeric enzyme. If the enzyme contains only one single polypeptide chain, then it is called as monomeric enzyme. If the enzyme contains more than one polypeptide chain, then it is called as oligomeric enzyme. There are two categories of enzymes. They are intracellular enzymes and extracellular enzymes. If the enzymes act where they are synthesized, then they are called as intracellular enzymes. If the enzymes act outside the cells where they are synthesized, then they are called as extracellular enzymes. Examples, all digestive enzymes comes under extracellular enzymes. According to International Union of Biochemistry, enzymes are classified into 6 major classes. Each class represents the general type of reaction that were carried out by the enzymes of that class. Those are oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. First one, oxidoreductases. These are the enzymes that are involved in oxidation reduction reactions. Means these enzymes catalyzes the oxidation reduction reactions. Example, alcohol dehydrogenase. Second one, transferases. These are the enzymes which catalyzes the transfer of functional groups. That means they catalyze the group transfer. Example, hexokinase. Hydrolases. These are the enzymes that causes hydrolysis of various groups. That means they are involved in hydrolysis reactions. Example, lipase. Lyases. These are the enzymes which are specialized in addition or removal of water, ammonia, carbon dioxide, etc. Example, aldolase. Isomerases. These are the enzymes which are involved in all isomerization reactions, which means interconversion of enzymes. Example, ketoisomerase. Ligases. The enzymes which are catalyzing the synthetic reactions in which two molecules are joined together and ATP is used. The reaction catalyzed here is condensation. Example, acetyl-CoA carboxylase. Enzyme inhibitors. Inhibitor is defined as a substance which binds with the enzyme and decreases the catalytic activity of that particular enzyme. Inhibitor may be of two types, whether organic or inorganic in nature. There are three various types of inhibitions. They are reversible inhibition, irreversible inhibition, allosteric inhibition. Reversible inhibition. In this type of inhibition, the substrate binds to the coenzyme non-covalently, which means weak bonds. There are two types of reversible inhibitions. They are competitive inhibition and non-competitive inhibition. In competitive inhibition, the actual substrate and the inhibitor have structural resemblance. If substrate binds to the active site of that enzyme, then enzyme substrate complex is formed and the enzyme activity is carried out. If the same inhibitor binds to the active site, the actual activity of enzyme is not carried out. To prevent inhibitor binding to the active site, then substrate concentration have to be increased. Example, succinate dehydrogenase is enzyme and its corresponding substrate is succinic acid, which is in structural resemblance to malonic acid, which is a competitive inhibitor of substrate succinic acid. If substrate goes and binds to succinate dehydrogenase, then action of this succinate dehydrogenase is carried out. If in place of substrate, this competitive inhibitor goes and binds to the enzyme, the activity of succinate dehydrogenase is not carried out. Non-competitive inhibition. In this type of inhibition, addition to active site, there is also a site called as allosteric site. Substrate binds to active site and inhibitor binds to allosteric site. The binding of inhibitor does not interact with the substrate binding to enzyme. The inhibitor does not interact with the binding of substrate to active site. Example, heavy metal ions. Irreversible inhibition. In irreversible inhibition, the inhibitor binds to enzyme covalently, which is a strong bond. So, the activity of enzyme is not carried out. Example, iodoacetate inhibits the enzyme papain and glyceraldehyde 3-phosphate dehydrogenase. Allosteric inhibition. Site other than active site is called as allosteric site. Allosteric regulators regulate the enzyme activity.